What I'm going to focusing on today, I have this broad title of Advancing LCMS Analyses for Characterizing Human Exposure to Poly and Perfluoroalkyl Substances. So that's the general theme. Hopefully it'll be interesting for those of you in the audience. So I always like to start my talks, hold on a sec, with a thank you, because I am the captain of the ship, you might say, but not really. I can only steer the ship in the direction that my students and postdocs help me achieve. And so, and all the work that I'll be presenting is really primarily the work of my students and postdocs over the years. So I've listed a number of PhD students and postdocs and collaborators as part of this effort that have contributed to some of the work I'm going to be presenting here in Colorado. It is actually a nice day, but it's not quite warm enough to go out river rafting, but this is one of the things we like to do in the summer here in Colorado. What I will be focusing on is obviously poly and perfluoroalkyl substances. I will say things got pretty interesting last summer, and I think they will get even more interesting in the next few weeks as EPA moves on promulgating their proposed maximum contaminant levels for at least some PFASs in drinking water. But last June, they announced these lower health advisories of quite low concentrations. So PFOA 0.004 nanograms per liter, PFOS 0.02 nanograms per liter, and GenX and PFBS as well. And I'm sure we'll hear a lot more, and there'll be some discussion about how you can actually measure things that low. But that's not exactly what I'm going to be focusing on today. I'm really going to be trying to understand and talk about how we're characterizing human exposure to these compounds for which regulatory agencies are very concerned about very low levels of exposure. So, I think we're going to hear later about global issues, and I do like to put this out there. This paper that was published just, I think, last summer, talking about PFASs in rainwater. It was something that many of us knew was the case, that these are present around the globe in rainwater. And so, yes, they are out there. I think you're going to hear some great details later about exactly how they're distributed in the globe. I'm going to try and bring it in a little more local. But it does bring in this question of, if these chemicals are everywhere, what are we going to do? And so PFASs are ubiquitous in the global environment. I think that hopefully goes without saying for people who are working in this space. And you could just kind of throw up your hands and just walk away and say, well, they're everywhere. What are we going to do? However, if we're going to talk about treating water or treating food or making sure they get out of food, we have to keep in mind that we can't treat all the water and soil. And so how do we go about prioritizing things? What are the things that we need to do to prioritize so that we can reduce human exposure to these chemicals? And so, the answer to that question of what do we prioritize really requires some careful thought and analysis. And from my perspective, you really have to make sure you understand the full complexity of how people are being exposed to these chemicals and the relative importance of different exposure routes. So that's kind of what I want to focus on today. But first, I want to be very clear in what I'm talking about when I'm talking about PFAS. So 𝗀𝗃𝗁 et al. Defined these compounds as aliphatic substances with carbon-fluorine bonds, essentially. And there was a recent proposal or OECD definition that was put forth that was a little bit more broad in terms of what defines a PFAS. And to be clear, the OECD PFAS, there's a great work that's been done by Emma Szymanski saying this means that by OECD's definition, you would have 6 million potential different compounds. For today, I am going to focus on the 𝗀𝗃𝗁 et al. 2011 definition, which is mainly the cast of characters you're probably used to seeing, but does include some ether components, which I probably won't talk much about. But it's important to pay attention to what is defined as PFAS because the regulatory implications of this could be pretty significant. So speaking of PFASs, what are we talking about? Poly or perfluoroalkyl substances, poly and per or poly. You can take your choice of how you put that out there. But at the end of the day, what we're primarily talking about are perfluoroalkyl acids, right? So PFOS and PFOA being two of the most commonly studied. But it's important to keep in mind that polyfluoroalkyl substances as well. So what I'm showing over here is a polyfluoroalkyl acid. This is a 𝗀𝗏𝗃𝗃 sulfonate. And we care about these because they can have problems in their own right, but also they can be precursors to the more persistent perfluoroalkyl acids. And so that'll hopefully be a theme that comes through in my talk today. So if we think about the PFAS family tree, it's not just PFOS and PFOA. I was a part of a study a number of years ago where we discovered not just 40 new compounds, but 40 new classes or homologous series of compounds associated with aqueous film forming foam or HBLF impacted groundwater. And what this means, and I'm going to talk about HBLF a little more in a moment, when we go out and look at any one site that has been impacted by firefighting foam, you may see 100 to 200 different compounds present, depending if you look at the soil or where we see a lot more compounds as compared to the water. But it's not just about PFOS and PFOA, to be clear. And again, why we care about these polyfluorinated compounds is that they can form these perfluoroalkyl acids over time. So what I'm showing here are two structures of one on the left here would be a ECF or electrochemical fluorination derived a group of compounds, the sulfonamidyl ethanols. And on the right, this is a complementary, or I should say, an alternative way of synthesizing some of these compounds made via fluorotelomerization. And if they're made by fluorotelomerization, they can form things like perfluoroalkyl acids, such as PFOA, whereas the ECF compounds can primarily form sulfonates, but also in some cases form carboxylates. And we primarily care about those polyfluorinated compounds because of that potential to form perfluoroalkyl acids, but there are some cases where you might be concerned about the polyfluoroalkyl compound in and of itself. So I like to use this image. I'll come back to this several times throughout the talk. It's adapted from a paper that L.C. Sunderland's group published a number of years ago. And it really talks about the different ways that humans can be exposed. You can obviously be exposed directly from products. I think you'll hear about that later today. I'm going to focus on exposure kind of through the environment due to something called HBLF, and I'll touch more on that. But one of the things takeaways I want to pay attention to, or I want you to pay attention to, is that there is no really one ideal list of PFASs to look at because it really depends on the exposure pathway you are interested in. So if you're looking at consumer products, it might be a very different list of compounds that you look for perhaps as compared to what we might look for at these aqueous film forming foam impacted sites. So the different sources and pathways may result in very different levels and compounds that humans are exposed to. So I'm going to focus in on these HBLF sources because it happens to be a pretty significant source of exposure for many people throughout the US, primarily through contaminated drinking water, but it's not just drinking water issue. So aqueous film forming foam, or HBLF, and I say that, I've been saying that quickly for many years, so apologize if it sounds like something different, but AFFF is what I'm trying to say. This was actually a minor component of fluorochemical production. Most of the foam that was produced was primarily used by the military, some for other sources. These were and still actually remain complex proprietary mixtures, and people have discovered what's in them as a result of research, but it's not been necessarily publicly disclosed for a lot of these formulations. And the thing to keep in mind is that these PFASs were added to these aqueous film forming foams specifically for their properties, and they were present at percent or in some cases gram per liter levels. Okay, and so these are very highly concentrated stock solutions of the concentrate that then would be applied to fuel fires. They're only intentionally applied for fuel fires, but in some cases you hear about them being used for other purposes. And they were directly released to the environment, either into in an actual fire scenario or spraying onto grounds trying to train for these sorts of fires. And so it's important to keep in mind that these are very complex mixtures of lots of different chemicals. The other thing is that it's very highly concentrated. And so this is a quick calculation that really kind of astonished me when I first did it. And I've updated it to reflect the health advisories that were last summer. But these were sold often in 5-gallon buckets. And one 5-gallon bucket of one of the historical formulations of 3M light water had enough PFOS in it to contaminate the annual water used by almost 100 million people if that water was only allowed to have up to 0.02 nanograms per liter of PFOS in it. Because that had gram per liter levels, you do the math, you end up with that being enough PFOS for that level for about 100 million people using water for a year. Okay, so it doesn't take much. The other way to think of it is that if you often hear about people talking about Olympic-sized swimming pools. If you filled one U.S. Olympic-sized swimming pool with HBLF, it would have enough PFOS in it to contaminate 25,000 years worth of the drinking water for the entire U.S. population. Okay, and they produced multiple Olympic-sized swimming pools of this material. And it's not just that it's highly concentrated, but also that it's been released at a number of places around the country. There's probably some more up-to-date maps, but this is one from a couple of years ago showing the U.S. Department of Defense sites where it had been measured, and I think there's probably even more at this point, but also fire training areas or airports where we know that this material was likely used or released. It's really all over the U.S. and honestly throughout the world. There's lots of other countries that have similar issues to what we have here in the U.S. So when we're talking about human exposure, or even just measurement of environmental samples, people often ask the question of, which compounds should we worry about? Should I use the EPA method 1633 list? Is there a better list? And I like to think of this as, what is the scope of what you're trying to look at? And this was a great paper, and I'll try and keep the lead author on a lot of these papers in the bottom left-hand corners. This is 𝗀𝗃 𝗏𝗏𝗀𝗃's work when she was a postdoc in my lab. Looking at the targeted compounds that we typically do with the triple quad LCMS analysis versus that which you might see with a high resolution mass spec or using something like the TOP assay, the total oxidizable precursor assay. And what I want to point out is that different sources and pathways may result in different analyses. So in some cases, you may want to do GCMS instead of LCMS. Or you may want to do one of these more broad techniques. But for what I'm going to focus on today, we set our sights mostly in this space. So mostly looking at things that are amenable to the TOP assay and or high resolution mass spec. And that includes some of the targeted compounds or a lot of the targeted compounds that we care about. The main difference between a targeted compound is essentially having an analytical standard available for it. And more of those are becoming available every day. But there are a lot of compounds that there aren't analytical standards available for. And this is some examples of these HBLF-derived polyfluorinated substances that we have observed either in foam or at foam-impacted sites. And these are just some example structures. The thing to keep in mind is that they have these very complex head groups. They all have that perfluoroalkyl tail, but have these complex head groups. And in some cases, they are negatively charged. In some cases, they're positively charged, and in some cases they're both positively and negatively charged, or Zwitter ions. And so when we go out and look at an HBLF impacted site, you may only see 100 to 200 different compounds, but we can look for up to 1,400 different compounds because we are typically using high resolution mass spectrometry and looking for this broad suite. And if you're interested in this sort of application, I would strongly encourage you to go take a look at the NIST PFAS GitHub website, where they now maintain this list of compounds that you can look for when you're using high resolution mass spec. When you're doing this, I would encourage you to think about how you're going to use that information to identify the compounds. And this is another paper put out last year where we updated some of the criteria for identifying unknown compounds via high res mass spec to be more specific for PFAS. So this table is a little bit hard to read. I'm not going to go through all the details, but I want to make sure you pay attention to it and go ahead and look up the 𝗀𝗃 𝗏𝗃 paper on ES&T letters Particularly look at the SI. We put a nice flow diagram in the supporting information to help you figure out what is the right PCI or PFAS confidence and identification level for your unknown analysis if you're doing high-res mass spec. But again, I want to focus on what people are being potentially exposed to as part of an 𝗀𝗄𝗄 release. And I think one of the key things to keep in mind is what is released may be different than what people are exposed to because of how these compounds behave in the environment. So even though we found all those compounds in the 𝗀𝗄𝗄, it doesn't mean that they are necessarily present in someone's drinking water. And the real question behind this is, do all of those polyfluorinated substances matter that are present in the foam? Do they actually make it into people's drinking water? Are people being exposed to these compounds? Are we just looking at the tip of the iceberg? Or are we actually characterizing things fairly well by just looking for perfluoroalkyl acids in analyses? And the short answer is no. We need to go beyond the perfluoroalkyl acids, and hopefully I'll convince you of that today. So what I want to put this in the context, if you're not familiar with the ITRC, which is a group that maintains some training materials, and this is from a fact sheet they put together. You know, if we think about a release scenario where we have firefighting foam applied to a soil, it migrates down to the groundwater and is transported. You know, we know that some of these compounds are really strongly retained in these soils due to their interfacial surfactant properties. They really like the air-water interface. But we also think that there's a really important role of these, the transformation of these cationic and zwitterionic polyfluorinated compounds in the subsurface. But you know, the question is, does that transform all of them in the subsurface before they get to drinking water? But to answer some of these questions, and this will make sense for human exposure more in a little bit, but we wanted to try and understand release of these chemicals from soils. And so another former postdoc of mine did a study where we took some of these soils, we essentially collected them from the field, we ran artificial groundwater through them, and we looked for the release of these compounds from these soils and measured all of the compounds that came off, both in negative and positive electrospray ionization mode, and tried to identify them and the rates of the release and what was the different chemistries that we saw coming off these soils. But the first thing I'll point out is that in order to do that, in order to have an understanding of what could be coming off soils, we actually needed a novel extraction method to analyze for particularly some of these zwitterions in the soils. And this was work from another former PhD student, Dr. Anastasia Nickerson, where she developed this alternative extraction method to extract the PFASs from the soils, analyze them via ESI positive and ESI negative analyses, and try to get a comprehensive picture of what is present in these 𝗀𝗄𝗄 impacted soils. And for anyone paying attention, this is not the same as draft EPA method 1633, which is only a basic methanology extraction. This does involve an acidic extraction to help get some of those cations off. And why that matters is that when we apply this to different 𝗀𝗄𝗄 impacted soils, you know, we see that the perfluoroalkyl acids like PFOS are not always the most abundant. In fact, on the slide you hear for 3 different soils, you see in some cases these cationic and zwitterionic compounds. The ones that you can see in the ESI positive mode are shown in red. Those are ones that we see a very high abundance of them in many soils, and they can be very important in terms of PFAS mass in the soils. And if we're not looking for them, we're not going to find them. And this is why, you know, I really advocate for high resolution mass spectrometry analysis of these types of samples. So when we go back to Andrew's study where we were looking at release from soils, just to give you a sense, we saw 167 different compounds coming off of these 2 different soils. And again, all these ones in red are ones that you really don't typically see in conventional analyses. These are examples of what these release profiles look like. If you haven't looked at, it's kind of like a chromatographic separation in an LC column, but it's actually a separation coming off the column filled with soil. We see the longer chain compounds coming off more slowly than the shorter chain compounds for both sulfonates and carboxylates. But what you observe here on the bottom here is that some of these more complex chemistries, these sulfonamide derived chemicals, which are very abundant at some of these sites, they take a very, very long time to elute. Some of them 100 times as long to come off the soil as some of their anionic precursor, or the transformation products. And in particular, the bottom right here, I'm showing all the different compounds that have a perfluorohexyl component to them. So hexane sulfonate, the sulfonate, the C6-sulfonamide, and other precursors. Some of those take a lot longer to come off than the hexane sulfonate does. And if you were only looking for the hexane sulfonate, you would think by pore volume 10, there's nothing left. But when in fact there are a lot of these precursors still to come off the soil. And some of them actually don't come off the soil, even in 100 pore volume. So this is our mass balance effort on some of these soils. On one soil, we actually got a pretty good mass balance. There wasn't as much on this particular soil. But that bottom soil, a lot of those compounds, if we compared what eluted from the soil and what was present initially in it, a lot of it stayed in the soil. It still didn't come off. And when things did come off, again, the precursors are really important. So what I'm showing here is the cumulative mass of the different precursors as well as their transformation products. So PFHXS being the ultimate transformation product. And what you see here is the C6-sulfonamide swamps, the mass of the C6-sulfonamide in this particular soil swamps the amount of PFHXS that's there. And this is a compound that is not typically looked for in many analyses. But there are other precursors. It's not just the C6-sulfonamide that's important. And this kind of sets the stage for why we're looking for some of these compounds in potentially impacted drinking water supplies. So, and the reason why we're paying attention to them, I should say, is that if we're looking at this broad suite of polyfluoroalkyl compounds, we know based on literature, and this is a review that 𝗀𝗏 did, that they can transform to the perfluoroalkyl acids. And I want to point your attention to this center of the diagram here, where we see this very last step is the conversion, and for many of these pathways, is the very last step is the conversion of these sulfonamide chemicals to things like PFOS, or if it's a C6 version, to PFHXS. And so just keep that in mind as we go through the rest of this. So if we go back to this, this question, how are we being exposed to them from these 𝗂𝗏 impacted sites? These chemicals are moving through the soil, they're getting into the groundwater and being transported in the groundwater. And so we have this question, do we actually observe any of these precursors in people's tap water? So if we would go out and collect tap water, can we actually observe some of these compounds in the water? And the first person to answer this question was a visiting student from China at the time, 𝗀𝗙, who published this paper where we looked at this particular impacted water had very, very high concentration. So PFOS was 33 ppb, or 33,000 parts per trillion. But partially because that water was highly impacted, we actually observed a lot of different compounds in that impacted water. They're all shown here in terms of carboxylates and sulfonates and fluorotelomere sulfonates, kind of the normal cast of characters. But we also observed some of these polyfluorinated substances that I was just talking about, and these sulfonamides. I will mention we did look in ESI positive mode, did not see any of these compounds that we would only see in the ESI positive mode, but we did see these other anionic precursors present in this particular tap water. But it wasn't just Zin's work. 𝗀𝗏 𝗪𝗏 also went and looked at some water samples here in Colorado, and we observed some of these sulfonamides, not just the C6 version, in some of these impacted wells in the security fountain wide field aquifer here in Colorado. And interestingly, there wasn't necessarily a strong correlation, at least every one sample. Broadly speaking, yes, there is a relationship between what we see in terms of the sulfonamides and other compounds, but there are some differences. Not every sample that has the perfluoroalkyl acids is going to have high levels of the sulfonamides, but we do see those sulfonamides in these impacted waters. So getting back to human exposure, if these things are actually present in the water, does that matter? Do these precursors bioaccumulate? And so 𝗀𝗏 went to try and answer this question by partnering with J.M. DeWitt. And what we did was some whole 𝗂𝗏 mouse exposures. So essentially mice were dosed with 𝗂𝗏. And we wanted to understand if we measured the serum of the mice. So it's an important thing. We were only looking at the serum of the mice. Did we observe anything in the serum for these polyfluoroalkyl substances? And so we looked at that, and the short answer is, if we looked at the 𝗀𝗏𝗃, this is a bubble plot shown on the left. A lot of the precursors are there in the kind of mauve color. But if you look at what we actually identified in the mice that were exposed to the PFASs in the 𝗀𝗏𝗃, we actually don't see very many of those compounds. In fact, what we see are primarily the end products, the ultimate transformation products of these precursors. So these precursors are not accumulating in the serum. But we were interested in the fact that we saw levels relative to what was in the serum that were elevated, which got us thinking, are the mice biotransforming these compounds if they had a direct exposure to this 𝗀𝗏𝗃? And so quantifying these compounds and doing a bioaccumulation when you don't have authentic standards is really hard. So we had an alternative way, which was to calculate essentially a pseudo BAF or bioaccumulation factor by dosing 𝗀𝗏𝗃 into serum to account for matrix effects, and then normalizing everything in our samples to PFAS, to linear PFOS. So the idea here is we try to account for potential matrix effects by normalizing to what's in a dosed serum, but also normalizing to PFOS to look at relative bioaccumulation. And what we observed here is that we saw, when we normalize it in these particular mice, we actually saw much more accumulation of PFH excess and heptane sulfonate. And this was, to us, very strong indications of precursor transformation, because we know that these particular firefighting foam that we were dosing with had much higher levels of precursors for these particular chain lengths, the C6 and C7, than it had for a lot of the other compounds. But we also saw, and there might be some indications for some of these other compounds that it was a precursor-driven one, but those were the two that we had kind of the most confidence in. But we also saw some of these other structures, the keto-substituted PFOS or the unsaturated PFOS, even though they were very low concentrations, much lower than PFOS itself. When we looked at the relative bioaccumulation, there was some indication that they have some increased bioaccumulation potential. And we don't think that in those cases it's a precursor transformation, but we think that those might be important ones to look at if you have highly exposed populations where these compounds are really abundant in, for example, drinking water. But the last thing I want to talk about in terms of human exposure is looking actually in human blood. And most work, including the work I was just talking about in human serum, or most work we do is focus on serum analyses. So where there's human serum or mouse serum, that's what we typically look at. And so it's relatively easy to analyze. You don't have to worry about whole blood cells, and it's different from whole blood, red blood cells. Is different from whole blood because you separate out those red blood cells. And collecting serum usually requires a phlebotomist. And it turns out, because some of these compounds very strongly bind to serum, you can actually get enrichment in serum relative to the whole blood, because the serum proteins are where some of these compounds bind. And so historically, a lot of folks have looked at serum for measuring PFASs in human blood. That's kind of the standard. But more recently, there's been some interest in this idea of looking at whole blood. And in fact, one of our next speaker, who I think can answer any of the hard questions about some of these efforts, came to me, was it 2 or so years ago, I think, maybe 3, and said, hey, we've got this technique we want to try out using volumetric absorptive microsamplers, or VAMs, which are shown here. It's basically a sampling device that will pick up a very precise volume of whole blood. And if you basically do a finger prick and you collect the drop of blood on your that comes out of your finger onto these samplers, you don't need much of this because of advances in LC-MS to do a quantitative analysis of what's in whole blood. And so when Andrew approached us about potentially doing this, we decided we would try and evaluate this and evaluate whether PFASs measured in whole blood via self-collected VAMs are reliable, And a secondary question is, are we missing anything when we're looking at whole blood versus what we see in serum? Because there have been some hints in some studies that you will miss things if you look in serum, despite the fact that that some compounds accumulate in serum. So even though I've been focusing on 𝗂𝗏 impacted sites, I am going to shift gears slightly and talk about a different type of exposure scenario. And that's essentially a paper manufacturing facility in Michigan. And Ков𝗏 𝗀𝗏, a collaborator at Michigan State, is involved in this and actually is leading the cohort in Michigan. But at the time when we, this was actually, I think, in the middle of the pandemic or right before things got really bad. We had a cohort we were developing in Michigan associated with this paper manufacturing facility. So this is a different exposure scenario than 𝗂𝗏, but it allowed us an opportunity to look at what do we see in serum versus what do we see in whole blood, and are those two things strongly related? Is it reliable? So Courtney led this effort to recruit participants for the study. We ended up having about 50 kits or so, 53 kits for VAMs that we were able to analyze, and Andrew's lab at Eurofins did this analysis. You can talk to him later about the reporting limits, but reporting down to a sub nanogram per mil, both for both serum and the VAMs collected whole blood. This gives you a sense of how we did this. So the phlebotomist collected whole blood, and then the participant collected the whole blood via metra. So these are what we call capillary VAMs, because it's collecting the blood from the capillaries in their finger. Samples were sent to Eurofins. They did all of the analysis via online SPE and LC-Tandem mass spec. I will comment, and again, I'll defer the hard questions to Andrew if you have them, on the actual sample prep and all that sort of stuff. But the key thing here is both the whole blood and the serum were extracted and analyzed essentially in the same way. So the VAMs and the serum, equivalent volumes and extraction procedures were used, so that really the only difference is that in one case you had a whole blood sample and you had a serum sample in the other. And then obviously they were collected slightly differently because we had to separate the serum out in order to get it. So what did we see? What did we see in terms of this effort? And this is under review. We're hoping it'll be out relatively shortly, but we're really confident in how this works, so we're happy to share it here. The good news is that these two things, serum and whole blood, were strongly correlated. So over several orders of magnitude, this is this elevated exposure situation for all the different compounds that we saw at high detection frequencies. So I think it was this is just showing things greater than 50% detection frequency. A very strong correlation between what you saw, what we observed in serum versus whole blood. And what is also important to note is that the 1-to-1 line is that dotted line there, and the 2-to-1, which is what we typically observe for many perfluoroalkyl acids, we see about twice as much in serum versus whole blood, is actually a relatively good approximation in general for most of these compounds. But there are some slight differences compound to compound. But the short answer, and that's what's shown here, the slope factors, is these slight differences between the slope of what you see in serum versus whole blood. But the short answer is this was very reliable. It gave us basically the same sort of data and was much easier to collect as compared to the serum, and essentially just as easy to analyze. But again, I'll defer the analysis questions to Andrew. But what I thought was particularly interesting was this table. And this is one where you might be like, detection frequency table, kind of boring. But if we look at it closely, we see that, yes, most all the compounds we see in the serum, we also saw in the capillary VAMs. This is pretty consistent. And then your eye might catch this compound, fossa. This is a sulfonamide. This is one of the compounds that is an immediate precursor to PFOS in many of the transformation pathways, and very similar to essentially just a longer chain length version of the C6 sulfonamide that I was talking about earlier, and the other sulfonamides we observed in drinking water. And for fossa, there had been hints at this in the literature and some studies showing this, but I think this is pretty definitive when we did not detect this at all in the serum samples, but it was detected in half of the capillary VAM samples, and the reporting numbers were basically equivalent. And what this is saying is that these compounds are either associated with the red blood cells or clearly not associated with the serum proteins, because we would have seen it in the serum otherwise. But it really points to the fact that some of these compounds may be more abundant in whole blood. And so, I very much encourage folks to think about, when you're trying to do these human exposure studies, at least consider going a whole blood route, because I think you may see some different chemistries in the analysis, and try to expand the analysis to different compounds. Obviously some next steps here might be to look for the C6 type versions or other shorter chain compounds in the sulfonamide class in an HLF exposed community. And we certainly hope to do those sorts of studies in the future, just haven't gotten there quite yet. So with that, I want to kind of give you some concluding thoughts and hopefully answer any questions that folks have related to what we're doing. If we're talking about moving the needle on understanding human exposure to PFASs, presumably as a step towards preventing human exposure to PFASs, I have a couple of thoughts here. One is just recognize these compounds are ubiquitous in the global and human environment. But the chemistry of the different exposures are different. And you'll probably hear some different chemistries when we talk about cosmetics later today, or other sorts of exposure through things like food, whether it's fish or produce or something like that. And I think there is this question that which exposures are really important. And you can measure compounds in a lot of different products and different things. I would say things you put on your body, we should pay very close attention to. Things you put in your body, you should pay very close attention to. So things like water and food. But it's something that we need to pay attention to, and potentially you need to think about prioritizing different sources of exposure, because if you look in everything, you're going to find PFASs in almost everything if you search hard enough. And so what are the ones we really need to focus on? A second comment, more specific to HLF, because that's what I spend most of my time thinking about, these HLF exposed communities or HLF impacted sites. They're complex. We see a lot of compounds at these HLF impacted sites. Some of these make them into actual drinking water sources. It really kind of depends how close the wells are or the water intake is to where that HLF was released. In some cases, you can see a lot of the polyfluorinated compounds. If in the case of a site where you might have, just fractures in the bedrock, which are rapidly transmitting some of these compounds in the subsurface. But if there's a lot of progression and a lot of movement through soil and through the a typical groundwater aquifer, you might see a lot fewer compounds in the HLF impacted drinking water supplies as compared to what was released at the site. And I just want folks to be aware of that. And I do think that you should think beyond the 1633 method list, because a lot of the compounds that we think are maybe important in these HLF exposed communities aren't necessarily on that list. And the last thing is that, we think some of these compounds are present in HLF impacted drinking water supplies, and potentially even things like foods also impacted by HLF. And they may be contributing to body burdens. And so I think this kind of raises the question of how do we capture the full exposure of humans to PFASs? Is it blood is one indicator, but there are some compounds that you might be exposed to that you excrete rapidly, and you can't actually measure in blood. So there may be some other things we want to think about when we're trying to quantify human exposure. And hopefully I've given you a sense of how we're using LCMS and high-res mass spec to understand these human exposures to PFASs. With that, I will leave you with some images from beautiful Golden, Colorado, where I am right now. It's not quite the same view out my window at the moment,